Beleza,
Inglês, tá?
beleza,
De
beleza,
última,
beleza.
de última hora assim é legal, né?
Não, não, mas tá tranquilo, tá tranquilo.
Achei que tava subentendido. Okay, guys. So, good afternoon, everyone. We're about to start our weekly webinar on this immuno webinar series that we've put together here. And our guest today is Professor Daniel Gomes. Daniel is from the Universidade Federal do Espírito Santo. Daniel is graduated as a biologist, right? But had his graduate studies at Federal do Rio de Janeiro and has just come back from London, right, Daniel, for a long time working there and has really exciting data on senescent immune cells, which I think it's one of the very hot topics, right, on immunity, how we age, right, or how our immune response ages. And I think it would be great to hear your talk regarding infections and the senescent or aging immune system. Thank you very much for being available, and the word is yours. Okay.
Can you hear me?
Yeah.
Yeah. Okay. Yeah. First of all, thank you very much for this lovely invitation. It's such good pleasure to be here. It's such a good pleasure to have the opportunity to present the data that we have been working for the past few years, which is associated with senescent cells, and especially trying to understand how senescent cells can be associated with the pathology that we can see in patients with cutaneous Leishmaniasis. I'm going to divide this talk in two parts. The first part, I'm going to give a brief flavor about how senescent cells can cause pathology, trying to show the mechanism, trying to highlight the whole of senescent cells in terms of function. And secondly, I'm going to give also another flavor about how senescent cells can be associated with the disease, but especially the severe of the disease. And also, how can we use senescent cells to discriminate patients who will develop more severe disease from those ones that will develop a mild or will cure spontaneously. So again, thank you very much. It's such nice opportunity to share. But before I go directly to the data specifically, I would like just to highlight one thing that is quite important, especially for the Southeast in Brazil. I'm from Universidade Federal do Espírito Santo, as Thiago mentioned before. If you look in this tiny spot, blue spot, you're gonna find Espírito Santo. Espírito Santo is a state that exists. Normally, people doesn't believe it. Only Mineiros know that a little bit about Espírito Santo, but Espírito Santo exists. Believe me, we are squeezed between the biggest states in the southeast, São Paulo here, Minas Gerais, and not only southeast but Bahia as well. And we are very, very famous for something that also I would like to highlight. Beach, of course, if you are Mineiro or came from Minas, you know very well. We have amazing beets. We have amazing coffees plantation as well. But also, especially if you work with tropical disease, we are very famous as well for malaria, leprosy, dengue, leishmania, and etc. If we consider the population per cases, we are in the highest, or one state with the highest number of this population. For those reasons, I established my laboratory here at UFES, especially Núcleo de Doenças Infecciosas, And we have been working with different disease, but most of the disease associated with the skin pathology. So disease caused by bacteria, by fungus. But today I'll be more focused in Leishmania, which is the main disease that we have been working for the many years. This is the group that are involved with the research that I'm going to show today. But especially today, I'll be more focused about senescent cells, which is part of the cells that we are studying, especially associated with the immune regulation and immune pathology of Leishmania. Talking a little bit about Leishmania, especially about American Tegumentary Leishmaniasis. I'm not going deep in the disease, but I also would like to highlight some points. Leishmania is a disease caused by a protozoan that belonged Leishmania genus. These parasite can cause a broad of clinical spectrum. Here you can see a patient that developed the diffuse clinical form. But also, if you move across this clinical spectrum, you're going to see in another side a patient who developed the mucosal clinical form. They are completely different, especially in terms of immune system reaction, but also associated with the parasite load that we can see here. People or patients who develop the diffuse clinical form, normally we don't see a strong inflammatory immune response or a cellular immune response. Normally, these patients, they develop a very strong humoral immune response, which also is associated with a strong parasite load. But in another clinical form, in another side of the spectrum, we have more inflammatory disease here, especially in localized patients or mucosal patients. In those patients, we can see a strong inflammatory immune response, along as well with a very strong cellular immune response. So those ones, inflammation and cellular immunity, are very well associated with the pathology that we can see in patients. And these basically offer to us, especially people who works with immunology, the opportunity to understand how the immune system can contribute for the tissue damage and the pathology that we can see. Much more than the disease mediated by or induced directly by the parasite, especially because in those clinical forms, we don't see parasite or are very low parasite. Basically, all the pathology, all the tissue damage, all the destruction, the skin destruction or mucosal destruction that we can see and observe in these patients is due the intense and exaggerated inflammatory immune response. So that's the point that we would like to address. And in this context, especially in the context of the pathology, one thing that we are trying to understand is how the senescent cells can participate of this. If we go deep trying to understand the pathology signature, we can highlight two important things. The first one, the increased cytotoxicity or the increased cytotoxic activity, especially mediated by CD8 T cells and NK cells, by producing granzyme, perforin, is very well associated with the tissue damage and cell death that we can see in mucosal and skin lesions of the patients. Along with this, the inflammatory immune response, especially mediated by producing IL-1 beta, TNF-alpha, which also is associated with the inflammasome activation. It's very well, and it's another pathology signature linked with the tissue damage and linked with the lesions that we can see in patients. Many groups have been showing this for the many years, but again, one thing that we are addressing, bringing to this image, is more pixels, especially related to the whole of senescent cells. We are demonstrating especially NK cells and T cells mediating or helping this pathology process. All this information was already published in the papers here, highlighted by the blue. But also I'm going to show more data, more things that we are producing quite recently regarding the pathology and senescence. What about immunosenescence and what is a senescent cell? That's the main thing that we are working by now. So just to let you know, immunosenescence by definition means the phenotypic and functional changes that the immune system acquire, especially during the physiological aging. As we age, our immune system change, and these change are especially associated or nominated as immunosenescence. And this change is very well associated with the impaired ability to respond to antigens, and also associated with the susceptibility to infections. We can see this very, very easy in elderly population. They are more susceptible to infection compared with the young population as well. So, but one thing that is very important is chronic stimulation. Doesn't matter if this chronic stimulation will be mediated by parasite, by bacteria or whatever, or could be also an autoimmune disease or an inflammatory disease. These can cause a premature senescence of the immune system in an age-independent way. Basically, when we are infected chronically, we can have the premature senescence of our immune system, which also is associated with the things that I told before, especially susceptibility to infection. What is a senescent cell, especially? Senescent cell share some features, two features that I'm going to explain in detail. But when we are looking at a senescent T cell, we can use those characteristics to understand, to identify them. But basically, a senescent T cell is a cell that is in an age state of differentiation. If we go to the memory compartment, we can identify especially the terminal effector memory population. Those subset, or this subset, is the proper senescent population. They express several markers, but also CD57 and KLH1 are markers that is very well described in senescent population. They don't express co-stimulatory molecules such as CD27 or 28. They have short telomeres. They have a very decreased TLR signaling as well, and they express some MAP kinases, especially P38, P21, P16, which is associated with the functional capacity of this population, and also the cell cycling control as well. As I mentioned, the cell cycling control is one of feature linked with the senescent T cell population, but also other populations that are senescent as well. So normally a senescent T cell or a senescent cell, they don't proliferate. It's a cell that is very poor or has a very poor proliferative capacity, but it's a cell that is a very, very functional. In terms of function, a senescent cell is associated with a secretion capacity to produce cytokines, chemokines, growth factors, and proteases, which are very well associated with the chronic inflammation. And in another perspective, a senescent cell, especially NK cell and CD8 T cell, are very cytotoxic, much more cytotoxic than a non-senescent cell, which is very well associated with the tissue damage that we can see in different disease. So if we taking these information in account, inflammation and cytotoxicity, which will mediate the tissue damage, both are the main signature associated with the immunopathogenesis of cutaneous leishmaniasis, as I described before, especially in cutaneous localized and also in mucosal clinical form. So that's the point, that the senescent cells could be part of the pathogenesis or could mediate the pathogenesis observed in patients, which nobody showed before. So I will start the data, especially trying to link senescent population or senescent cells and pathology of the disease. Most of the data that I'm going to show here was data obtained using PBMCs or using skin biopsies from patients. These patients was attended here in Victoria in the university hospital. So in this university hospital, we took those patients, and after that we did different experiments using different approaches, especially transcriptomic analysis, histological analysis, and cell isolation as well, to perform functional experiment using these samples. As a control, in most of the experiment that I'm going to show as well, we used health aged met patients, not patients but donors, that was kindly obtained, and they accepted to donate PBMCs and also skin biopsies. And then we did all the things in comparison between them. The first thing that we did, it was a long time, not a long time ago, but this paper was published in 2019. We took blood first, and trying to understand the participation of senescent cells in infection caused by Leishmania. We got PBMCs, and then we did a pan characterization, especially associated with the senescent features. So basically, we isolated CD4, isolated CD8, NK cells, B cells from patients and controls, health controls. And we stained for telomeres, we stained for the markers that I mentioned before. And basically, we could demonstrate that the circulating T cells, CD4 and CD8, they accumulate senescent features. So they had short telomeres, they didn't express co-stimulatory molecules, they didn't have a telomerase activation, they didn't proliferate as well. They expressed most of the senescent markers, the main senescent markers for receptors, DNA damage. But just summarizing, two things were very important here. The first one, when we activated these cells in vitro with the Leishmania antigen, they showed an increased propensity to migrate to skin, especially because they upregulated the CLA activation as the cutaneous leukocyte antigen, which could potentially impact in their participation in lesions. Secondly, we correlated the lesion size of patients with the different subsets that we can see into the memory compartment. So here is a naive population. From here, if you move across to this, you can see here for the less differentiated subset to the highest differentiated subset. And basically, most of the differentiated subset represented here by effector population or terminal effector population, both correlated with the lesion size from patients. This is CD8 T cells, but also we saw the same thing happened with CD4 population as well. Basically suggesting that the senescent or highest differentiated population could migrate to skin and could be in skin mediating the pathology. This is blood. This is not skin. We needed to show this in skin as well. And then we decided to analyze the skin, as I mentioned before, and we took the biopsies to do transcriptomic, to do different experiments. We start showing the skin experiment by analyzing the transcriptomic signature that we identify in lesions. Here is the gene ontology highlighting the whole cell process that we could see in lesions. And also I would like just to highlight here, aging and replicative senescence were some cell process that were more expressed in lesions at the time. Just to certify this, we took senescent K gene signature, especially associated with genes that control the senescence process in the cell. Here we can see P21, P16, which controls the cell cycling and proliferation. P38, which control the production of pro-inflammatory compounds. Cestrin control the stress and different also process in the cell. ATM, ATF is the DNA damage repair machinery as well. Gomes is a transcriptional factor associated with the senescence, and TBET is associated with inflammation or functionality in senescent cells. And basically, all of those genes, they were upregulated in lesions, represented here by the red bar, and we didn't see these in health skin, suggesting to us again that the senescence were there taking place, doing some important thing. We did the convolution after that. We identify different population in lesion. So, and highlighting here, we have more differentiated population represented by the effector memory T cells or TEMRAs or terminal effector memory T cells, or NK population that is more differentiated, correlating positively with the senescent genes. So again, suggesting the whole of senescence signature, and especially linking these signatures with the most differentiated population. So cytotoxicity and inflammation are by now or so far are the main signature linked with the pathology of the disease. So one thing that we decided and we tested was associate the senescent features with those ones, trying to see how senescent could be linked with the cytotoxicity and inflammation. Just to let you know, the senescence score positively correlated with the inflammation score and with the cytotoxic score as well. So this suggested that they could be linked. And just to identify, especially the genes linked with senescence during those processes, I don't know if you can read here clearly, but I will let you know. We took genes linked with the secretion capacity of senescence. We took genes nominated by SASP. SASP is a senescent-associated secretory phenotype. Basically, this phenotype is composed by pro-inflammatory cytokines, pro-inflammatory chemokines, some proteases as well. And we identified those compounds upregulated in lesions, which were completely different from health controls. Health controls are in these blue bars, and patients are represented here under these red bars. You can see here, most of these genes they have a completely different pattern in comparison with the health skin. But most importantly, these genes, they strongly correlated with the late or more differentiated population, or senescent population, or more senescent population than other subsets. The same thing was observed when we analyzed the genes linked with the cytotoxic capacity. So again, here you have patients and controls under these blue bars. Most of the cytotoxic genes, they were upregulated in lesions, but not in health skin. And again, they correlated with the more differentiated population. So this data was very important to us to demonstrate the whole of senescence, or the association of senescence with lesions. But this is the transcription factor, this is RNA. So we needed to verify the participation of senescent cells in lesions properly. To do that, we took the biopsies and then we stained for NK population and CD8 population here, represented by the green markers, the green staining in these immunofluorescence. We stained as well for cytotoxicity, and we stained for CD57, which is a senescent marker. It's a very well senescent marker. And basically, we could see both population in lesions. Here is granzyme B positive cells, CD57 positive cells. We saw an accumulation of CD8 higher than NK in lesions. Lesions also, of course, they had much more cytotoxic cells. It doesn't matter if it's NK cells or CD8 compared with the health control here. But one thing that also highlighted our attention is when we correlated the lesion size of patients with the frequency of these senescent population, we could see in both cases an association, a positive correlation, but especially CD8 population, they correlated much higher than NK population. This confirmed to us the participation of senescence, or the senescent cytotoxic cell in lesions. So another question that remained was how this population, or how this senescent population could mediate the pathology. I mentioned that they are very inflammatory. I mentioned that they are very cytotoxic, but especially how a senescent cell, NK cell, CD8 T cell, could mediate the pathology directly, causing the tissue damage. Just before going to the result, I would like just to highlight this important thing. This is a differentiation profiling of T cell. This can be a senescent CD4 or senescent CD8 T cell development diagram. So we have here the early differentiated population moving to a terminal differentiation, or a senescent feature. As the cell go toward senescent, they increase the capacity to express NK receptors. And this is very, very important, because also we can use the NK receptor, such as NKG2D, NKG2C, to identify the population. In the same time that they increase the capacity to mediate cytotoxicity. In opposite, this population, they reduce the TCR signaling, which is very bad, especially to do the antigen-specific immune response. This is very well known, especially associated with autoimmune disease, chronic inflammatory disease, aging, and Leishmaniasis infection. This was demonstrated in mice, an accumulation of NK cells, CD8 T cells expressing NK, and this is called the bystander cytotoxicity, when the cell do the cytotoxicity mediated by NK receptors and not by TCR receptors, recognizing MHC class 1 or class 2. So we have demonstrated a couple of years ago that the senescent T cell, especially CD8 T cell, but also this was showing CD4 T cell, they have a very strong identity of NK population. If we compare a naive population, which is a very early differentiated population, with a senescent population, a true senescent population, they are completely different in terms of NK signature. And by single cell analysis, we have demonstrated as well that these differences are linked with NK receptors, such as the NK receptors, NKD2D, NKD2C, CD57, as I mentioned here, demonstrated by the red bar, which is linked with the terminal differentiated populations, compared with the early differentiated population here from central memory or effector memory. Also, a senescent population has an increased identity of chemokines, cytotoxicity, NK molecular adapters to activate the NK cell function, and they decrease the TCR signaling as I demonstrated before. This was done using cell from health donors. So, and this was mainly linked with the CD8 T cell. I told you about the CD4 T cell. CD4 T cell can be senescent and can go towards senescence, especially expressing all the features that I demonstrated before. But historically, CD4 in Leishmania infection, they are very well associated, especially with the protective immune response, especially by triggering the macrophage to kill the parasite by producing ROS or nitrogen species as well. But one thing that is very well known, especially in virus infection, is during chronic activation, the CD4 can be reprogrammed, and this reprogrammation is very well associated with the acquisition to mediate cytotoxicity. Basically, CD4 under chronic stimulation can be a proper cytotoxic cell and also can express senescent markers, as I mentioned before, here represented by KLH1 and by CD57. One thing that arises was, does CD4 or the cytotoxic CD4 T cells could be associated with the Leishmania pathology as well? So just to answer this question, we took the biopsies and we did a multiplex analysis. We set a multiplex facility here, a digital pathology facility here in Espírito Santo. We are doing up to 30 markers in tissues, in different kind of tissues. Here we have a cutaneous leishmaniasis lesions stained with 21 markers. We did a staining for different cell types, cytokines, cytotoxicity, but highlighting here, we could see CD8 and CD4 T cells expressing granzyme. So we just confirmed this in a single area analysis. We can see here CD4 highlighted by yellow or CD8 by green, producing granzyme highlighted by red. And then when we analyzed the cytotoxic cell expressing an NK receptor, as I mentioned that the senescent population can express NK receptors, we also could identify both CD8 and CD4 in lesions expressing NKD2D, but also we have NKD2C as well, and also other NK markers. So this population was very well distributed in the lesions, and also just to certify that they could do the killing and that they could mediate the killing, we also analyzed the ligands for these receptors. So we analyzed by transcriptomic here. You can see the HLE and MIC-A and MIC-B all are ligands from NKD2D and NKD2C. They were upregulated in lesions. And also we identify cells that was expressing these ligands. So we identify macrophages, monocytes, also identify fibroblasts expressing those ligands in the lesions. Just to confirm this, just to confirm if the cytotoxic CD4 or CD8 T cells could be interacting with monocytes and fibroblast or macrophage, we stained again, for all those population, and we analyzed the cell interaction. Here you can see the different population in the same area, CD4 in orange, CD8 in green, monocytes or macrophage in dark blue, and in light blue was a fibroblast. We can see here they are very close, and this analysis is a cell-cell interaction. Basically, when we have this Dutch, means that the cells are interacting very, very close, less than 5 micrometers of proximity. So those ones show to us that they were interacting, and they, especially monocytes and fibroblasts and macrophage, they could be potentially being killed by this cytotoxic population. So just to go deep to the CD4 cytotoxic population and demonstrate that this cell could especially be a cell helping the pathology, because we know that CD8 does cytotoxicity, and the CD8 is very well associated with the pathology. But what about the CD4? CD4 was a bit surprised to us, especially mediating the killing in lesions. We took biopsies from the patients, and these biopsies we did the phenotypic and functional analysis. Also, for the analysis we used the blood as well. I'm just going to show a couple of analyses, especially linked with the flow cytometry directly from the biopsies. We didn't do anything stimulating. We just took the cells, we pulled out the cells, and we stained for granzyme, we stained for different cell populations. We could see, of course, NK cells in lesions, CD8 cells in lesions, but again, just confirming the things that we saw in the immunohistology, we could see CD4 expressing granzyme in lesions, but the CD4 from blood, in patients, they don't express granzyme, suggesting that something that was going on in lesions promoting the cytotoxicity in the CD4, or maybe helping the gain of cytotoxicity for all the cell population. So this NK population also, they express the NK receptors, as we saw in the immunofluorescence. They express NKD2D, so the lesional NK cells, they express much more NKD2D, much more NKD2C. They decreased the expression of NKD2A, which is a inhibitory receptor, and also they increased the CD57 receptor compared with circulating CD4 isolating from the blood of the patient. So lesional cells, or especially lesional CD4, they retained the NK phenotype, and also they retain the more senescent phenotype. Just to confirming this, we stained for the memory compartment as well. Here we have cells from blood and cells from skin. You can see here an accumulation of late differentiated population in skin, much higher than in blood from the patient. And when we look at the expression of these receptors, the NKD2C, the NKD2D, and the CD57, in these cells, especially the lesional cells, we can see that the proper senescent population here represented by EMRA, it's a population that retain the most of the NK signature, and also the most of the senescent signature compared with the earliest differentiated population. Also, the cytotoxic capacity was much more associated with the EMRA population, with the senescent population, represented here by the granzyme B production capacity and the CD107 capacity compared with the other population. And then we just to confirm if they could do the proper killing assay, we took different cell populations, CD4, CD8, NK, and we did a cell culture with a cell line called K562. It's a lymphoblast cell that don't express MHC class 1 or class 2. So this population can't be killed by MHC, by TCR way. So most of the killing mediated, they use NK receptors, and surprisingly and good to us, we could identify that the lesional CD4 kill in the same fashion way that CD8 and NK from lesions, but also from blood. But blood CD4 T cell could do the proper killing as we saw with the other cell type. These data really show to us or confirm to us that the lesional environment retain the senescent population, and these senescent population could be the most important population linked with the pathology. Just to take the message, the whole message about this study so far, we just showed that the accumulation of senescent cells in lesions from CD4 and CD8 compartments, they potentially assist, especially CD4 T cells. They could assist the lesional pathology by triggering target cells through NK receptors, NKD2C or NKD2D. I didn't show, but we did also experiment blocking these receptors. And when we blocked, so we reduced the killing capacity of these cells. So this will be the next paper that is in press as well. And also we showed that this bystander cytotoxicity is mediated by a proper senescent subset. All this data was very, very fresh, published maybe 15 days ago in Frontiers Immunology. I can also give to Thiago the information about the paper and the details about the paper, and go very, very fast also about the whole of senescent cell, especially mediating or using the senescent signature to predict the clinical outcome. Also, I would like to highlight another fresh data that was very, very published also 15 days ago, very soon, which is associated with the senescent signature. So I'm giving a spoiler about the data that I will show, but using the senescent signature to predict the disease outcome, and especially when we look at the localized and the mucosal clinical forms. We don't know in clinics what's going on with patients who develop the mucosal clinical form. The clinical form is about 2 to 5% of the patients who developed the localized disease first, but just 5% can progress to develop the mucosal clinical form, and we don't know what's going on in these patients, what is the things associated with this progression. So basically, we took the biopsies from these patients. We did transcriptomic from diffuse, localized, and mucosal. So this is just to highlight that we could see completely separation for the clusters. So health controls, patients with diffuse, patients with localized, and patients who progressed to mucosal. It's not patients that had mucosal, but patients with localized disease and then progressed to mucosal clinical form. So you can see here the genes expression pattern. They shared some genes, but also we can see genes that are very only expressed in each clinical form. And these genes were very strongly linked with inflammatory immune response, skin healing, but also would like to highlight the senescent genes. So when we analyzed the expression of senescent genes, the same genes that we did in cutaneous leishmaniasis, we could see a differentiation pattern in terms of expression, especially compare health skin, diffuse skin, and with other inflammatory clinical form represented by mucosal and localized ones. But one thing that I would like to highlight is most of the senescent genes were very, very linked with the localized and the mucosal ones. But most importantly, most of the senescent genes, they were higher in mucosal progressors compared with the localized patients. So this we could see for P52, P53, P38 as well, P16, Cestrin2, ATF. Most of the genes linked with the senescence, they were higher in mucosal clinical form or progressors for the mucosal clinical form than other clinical. Just to confirm this, we took the biopsies from patients, from lesions, and we stained for some genes. I'm showing just two, P53 and P16 as well. And we can see here in the mucosal patients, they really have more expression of all those ones that I highlighted in transcriptomic, but in lesions as well, they were higher in mucosal clinical form compared with the localized clinical form. Okay? When we look at the other pathology signature, as I mentioned before, we know the inflammation, we know cytotoxicity. It's another signature linked with the pathology, especially for the mucosal and the localized. We can see here that the signature or the score for this signature are completely different between the clinical forms. Again, especially cytotoxicity were in senescence, they were very strongly associated with the localized and the mucosal progressors compared with the diffuse or patients health controls donors. But again, most importantly, only senescence signature could discriminate between mucosal progressors versus cutaneous leishmaniasis. This suggested that the senescent could tell us something especially related to the progression of the disease. Just to confirm this, we did a ROC curve using the data set from the mucosal progressors, and we could identify that the score for the pathology signature, they could be useful to discriminate the progression the progression of the disease for the mucosal development, but only senescent could discriminate up to 90% the patients who will progress to the mucosal clinical form. Cytotoxicity, we could see around 75%, and taking the inflammation signature was very bad to discriminate the progression to mucosal clinical form. From the genes linked the senescence, we saw also different genes that were associated with this prediction, but highlighting, I would like to highlight P16 and P53 that predicted almost 100% the progression for the mucosal clinical form in those patients who had at the time the cutaneous clinical form, suggesting that the only senescent could be used, but between the senescent genes, P16 and P53 could be very, very useful signature to discriminate the senescent, the severity of the disease. So just to finishing the presentation, I would like to highlight the second part. It's like the senescent signature is found in other clinical forms, and they operate very differently between the clinical forms that we can see. The senescence is the most representative pathology signature of the disease, and also they could be useful to predict the disease worsening, especially linked with the mucosal development. But also, I'm not going to show today, but also the senescent signature can predict the clinical fail as well in patients. So that could be very, very useful for clinics to track the patients, especially for treatment reasons. So yeah, that's the thing that I would like to present today. I'd like to thank you, the group, and especially as well Professor Luiz Felqueto, who is the clinician involved in the patients, the sample collection as well. Several collaborators around the world also were involved in this work, especially Professor Arne Akba, Professor David Moser as well, Arne from London, David Moser from Maryland University, some collaborators, Professor Edgar Carvalho, Valéria, Cláudia Brodsky, Lucas Carvalho, Herbert from Fiocruz, from Rio University as well, and also the agents that supported the work, especially FAPES, who is the foundation from Espírito Santo State, CNPq, MRC, yeah, and I'm very happy to address the questions that can arise. Thank you very much.
Very nice, Daniel. Thank you very much for the presentation. It's always good to hear your work.
Amazing.
Can I stop the
yeah,
sharing?
You can stop sharing.
Yeah,
sure. Very nice. So I'm going to ask for the students, we always open the questions first for the students. I'm going to ask them or to open up the mic and talk directly to Danielle, or write to us here in the chat so we can read. I'm trying to see here. I didn't check the YouTube if there are questions there. I'm just going to do that. None, I think, till now. So, anyone? Questions for Danielle?
I'm just gonna ask a very quick question. Danielle, first of all, it's excellent work, really, really amazing. In your CD8 cells, do they have like a bias toward one of these subtypes of effector cells, like TC1, 2, 9, 17, 22? Is there any, or is just a general mix? Did you check for that?
That's a very good question, Gustavo. No, we didn't check. Now we are doing more deep analysis in terms of cell subsets phenotype. We're phenotyping for gamma delta T cells, for different everything that could be linked with the cytotoxic capacity. We are doing in lesions. Especially using the cell dive, the immunofluorescence, trying to understand specifically the whole of different subsets in this context. But no, we didn't so far, but very soon, I hope so.
Right, good.
Questions for Danielle, please? While they decide themselves, Danielle, just let me
Podem falar em português também, não? Se eles, às vezes eles estão tímidos.
Claro.
Pode falar, mas essa turma eu vou te dizer, nós vamos ter que, assim, para cada seminário, vamos ter que escolher 4, 5 alunos para fazer.
Valendo nota? Gustavo?
Lendo assim, se fizer bem feito, tá ok. Se fizer mal feito, perde nota. É simples.
Isso, ótimo. Então não
Valendo
ganha, só
tirar
perde,
nota.
né?
Não, valendo
Só
manter a
perde.
nota, né?
Mas, Daniel, deixa eu, então, só para quebrar o gelo dos meninos aqui, deixa eu te falar, eu sei que você, né, o seu foco é sempre ter e tudo mais, mas assim, se chegar a olhar via inata, TOL, inflamassoma, alguma coisa, tá, isso funciona igual? Ou dentro desse contexto que vocês estão colocando de senescência, assim, eu sei que você está falando de uma senescência muito específica, mas assim, você vê algum distúrbio no começo, do outro lado da ponte?
Então, essa também uma pergunta muito boa, Tiago. O que que acontece? Até hoje tem uma discussão muito grande dentro do contexto do grupo da senescência se célula inata de fato adquire características de senescência. Então, por isso que ninguém nunca olha muito com os olhares da senescência para a célula inata. Ela expressa algumas coisas que poderiam ser indicativos de senescência. Mas também em outros casos, por exemplo, P38, que é um baita de um marcador de senescência em linfócito, a célula inata ela expressa o P38 para produzir TNF-alfa, enfim, que qualquer coisa vai fazer com que ela seja produtora, né? Então, assim, existe uma dificuldade muito grande de caracterizar a resposta inata como senescente. Por exemplo, a questão de proliferação celular não é tão impactada, por exemplo, em célula dendrítica, enfim, em outros compartimentos da inata. Esse é um ponto importante. Então, um prêmio de US$ 1 milhão, o cara que de fato conseguir discriminar é, as- a, a própria característica de senescência no compartimento da inata. Mas tem um ponto importante na adaptativa, porque a senescência ela está ligada com essa capacidade de responder de forma antígeno inespecífica. E célula senescente expressa TOL pra caramba, cara. Se você pegar no compartimento terminal de diferenciação, a resposta de TOL ou via TOL, né, é uma forma muito grande de resposta dessas células terminalmente diferenciadas. Tem um paper que saiu um ano passado, cara, na, na Nature, de um grupo lá da UCL, que eles mostram célula T residente hepática expressando TOL e o reconhecimento de bactéria que vem no sistema porta-hepático para o fígado, sendo um importante mecanismo de imunorregulação, mas mediado por célula altamente diferenciada, por célula senescente lá no fígado. Então, eu esqueci de falar, né? Eu estou falando da célula senescente no contexto de uma patogênese,
Claro.
mas a célula senescente, ela não é ruim o tempo todo, tá? Ela é ruim em alguns casos, nos casos onde a gente tem uma inflamação exacerbada, né? Num estímulo crônico. Mas do ponto de vista fisiológico, a célula senescente é a célula que vai fazer esse controle, né, de bactéria que chega no fígado para não causar a treta, é a eliminação de célula tumoral, por exemplo. Então, assim, a célula senescente no dia a dia ela é ótima, é bom ter. O problema é justamente quando você tem essa resposta inflamatória exacerbada. Eu respondi o seu e acabei misturando com uma outra coisa, né? Mas
Não, não, isso aí
nesse
mesmo.
contexto aí,
Não, mas
tá?
assim, eu vou te dar o contexto também da minha pergunta, cara, porque assim, a gente sabe, teve aqui, inclusive, recentemente, eu não lembro agora se semana passada ou anterior, uma palestra sobre trained immunity, né? Que
Aham,
é
aham.
justamente ao contrário, né? É sobre a memória, entre aspas, de célula inata, de modificações epigenéticas que geram. Então, assim, na realidade, são vários tons de cinza, né, Daniel? Não tem preto e branco, né? Então, acho que
Não.
do mesmo jeito, tinha um paper de uma década atrás, mais ou menos, que eu gostava sempre de passar para os alunos para lerem. Papel, se eu não me engano, de mídia 88, linfócito T, e tinha muito a ver com migração celular, entendeu? Não tinha muito a ver com a capacidade de resposta, mas a capacidade de responder ou migrar frente a estímulos de miocinas e tal. Enfim, eu estava perguntando mais dentro desse contexto se
Não.
esses mediadores não estão.
Mas é ótimo, cara, porque assim, a gente aprende imunologia no ABAS, né? E no ABAS, nesses livros de imunologia aí. Cara, os caras engessam para ficar didático justamente isso, que linfócito expressa o TCR, que macrófago, célula inata expressa os receptores, né, os TOLs, enfim, barará. Mas, cara, a gente sabe que não é isso que funciona, né? Que se tem essa plasticidade e em cada contexto, em cada doença, em cada tudo, essa plasticidade faz com que as células se comportem de uma forma, de outra, seja NK aqui, seja. Então, eu acho que esse tipo de abordagem que você está falando é muito legal, porque abre essa possibilidade dos alunos verem, poxa, o linfócito expressa TOL pra caramba. Não é todo linfócito, nem em todo contexto que você tem essa expressão maciça de TOL. Né? Em alguns contextos, e isso é importante passar para a moçadinha. Mesma coisa da modificações plásticas, né, pelo training immunology, né, modificações epigenéticas, etc., que direcionam as funcionalidades de células adaptativas, né? Então, assim, é muito legal para as pessoas entenderem e não se assustarem com uma coisa que já está publicada há 30 anos, né? Porque às vezes você pega um artigo assim e fala assim, uau, linfócito expressa TOL. Pô, mas se você pegar ali, isso já tem 20 anos que já saiu o paper mostrando que linfócito expressa TOL. E aí a gente ainda se assusta com umas coisas dessas, assim, que não tá tão oculta assim mais na literatura. As pessoas, quando eu comecei a mostrar esse negócio do CD4 citotóxico, o pessoal ficou assim, meu Deus, você tem certeza que essa célula... Primeira coisa que perguntar, né? Você tem certeza que essa célula é CD4? Você fez o sorting de verdade para CD4? Eu falei, lógico que eu fiz o sorting. Assim, contesta muito mais as questões técnicas do que, de fato, a capacidade da célula ser plástica e de fazer citotoxicidade. E aí, o Gustavo que trabalha com vírus já sabe disso há muito mais tempo, cara. Que isso foi mostrado em vírus, ó, desde que minha avó era criança, né? Que CD4 sendo citotóxico. Não surpreende muita gente. Eu fiquei um pouco surpreso na lesão. Mas muita gente se surpreende mesmo. Fala, nossa, CD4 fazendo citotoxicidade, eu nem sabia. Então, porque lá
Mas
no Abas mostra isso, né? Justamente isso, CD4 é MHC de classe 2 helper. E não é assim que funciona, né?
é verdade, mas aqui também tem muita coisa que foi tecnicamente mal feita e que acaba ficando preservado como se fosse um fato, e
De
depois
fato real, né? Não.
alguém vai lá e contesta, né? Então, acho que tem as duas coisas. Agora, eu ia aproveitar de perguntar na verdade, uma outra coisa, assim, mais talvez, não sei se é filosófica, mas a gente consegue, você imagina que a gente conseguiria, ao entender melhor a célula senescente, aplicar esse conhecimento para fazer com que uma célula CD8 normal ficasse mais poderosa? É uma questão. A outra questão é talvez inversa. Será que a gente consegue fazer uma célula ser mais senescente ou ter algumas células desse tipo no microambiente tumoral, por exemplo, onde você quer causar uma certa destruição? Como é que você
Não,
pode aplicar essa coisa em duas situações, em dois contextos?
cara, baitas perguntas. Existem, na verdade, abordagens reversas, tá? Você está falando de promover a senescência para as questões tumorais, por exemplo, sabendo que ela poderia ser uma célula mais
Por
citotóxica.
isso que eu falei, eu nem sei senescência, né? Promovendo
Sim, mas
alguns
com os
aspectos,
aspectos.
né?
É, Isso é uma coisa que a gente vem fazendo agora, o interesse até da gama delta, é porque a gente tem visto que a gama delta ela tem essas características, e a gama delta já tem sido usada nesse tipo de abordagem. Então, assim, a gente precisa só confirmar se de fato a gama delta tem ou é uma população senescente ou que carrega isso, e aí isso seria a resposta para a sua pergunta. O oposto também tem sido feito. Em situações de inflamação crônica ou mesmo nos idosos, para você potencializar uma resposta protetora, já que o acúmulo de célula senescente impacta muito na manutenção daquele ambiente inflamatório crônico, que está ligado também à incapacidade de responder a antígeno vacinal, bloquear ou eliminar a célula senescente com drogas que a gente chama de senolíticas, né? E já tem drogas no mercado, inclusive. Tem uma droga da GSK que foi até usada num trabalho do Arne, é, onde ele fez um teste humano mesmo, deu essa droga, tablet, os caras inibiram a P38, era um inibidor de P38 por 2 dias e foram vacinados. E os aí que receberam esse inibidor responderam muito melhor aos antígenos, né? Enfim, aos antígenos da vacinação. Então, os dois pontos eu acho que são muito possíveis. Mais possível agora, ô Gustavo, eu acho que é a questão senolítica. Primeiro, com apreço estético, porque quando a gente fala de senescência, todo mundo pensa em não ficar velho, né? É um negócio muito engraçado isso. Acha que é a fonte da juventude, mas estética, né? Não a fonte da juventude de viver muito tempo bem, né? E sim se viver bonito, por pouco tempo, talvez. Atacando de fato essa questão do envelhecimento de fibroblasto, né? Das funcionalidades estéticas. Tem vários grupos que vêm trabalhando com isso, com inibidores diversos, ainda experimental ou ainda em fase 1, fase 2, mas pensando na abordagem terapêutica, eu acho que seria possível também, muito factível mesmo. O gama delta já sendo uma coisa empregada, mas se a gente conseguir mostrar que de fato o gama delta tem essas características que a gente já vem mostrando que alguma coisa está associada, eu acho que sim, tá?
A
Maravilha,
Cris,
obrigado.
a Cris Bandeira tem uma pergunta, ela vai ligar o microfone. Cris, a palavra é sua.
Eu tenho, obrigada, tia, ao Daniel, linda apresentação, dados impressionantes, obrigada pela apresentação. Eu tenho uma sugestão, uma curiosidade, na realidade uma dúvida, não sei exatamente o que que é. É o seguinte, mas você estava mencionando isso agora há pouco. Você não acha que seria interessante fazer uma análise do tecido, porque todas essas análises de senescência, dessa última parte do seu trabalho, que você fez e mostra muito elegantemente como ela pode ser preditiva da virada para a mucosa, né? Para a virada da forma mais grave, né? Você fala disso. Mas
Uhum.
não seria interessante saber se o tecido não lesionado desse mesmo paciente tem a mesma assinatura? Você entende? Um tecido do. É porque toda
Vou te
aquela
responder, Cris.
uma
A gente
amostra
tem
da.
esse dado já. A
Já tem
gente
esse
tem
dado?
biópsia, a gente já tem esse dado, eu só não mostrei. A gente faz a biópsia da lesão e faz uma biópsia do braço ou a perna ou do membro contralateral. E não tem. O membro contralateral, ele tem a mesma assinatura de uma pele saudável. Ele não
É
tem
mesmo?
ele não tem esse acúmulo de populações senescentes. Uma coisa que a gente vem fazendo agora, além das populações que eu comentei com o Gustavo, tentar entender como que o ambiente lesional está impactando nessa indução de característica de senescência, porque é local, não afeta outro compartimento tecidual do paciente que não seja aquela lesão, tá? Não deu tempo de mostrar isso.
O compartimento sanguíneo tem algum impacto, né?
Tem um
Se eu
impacto,
lembro.
é bem pequeno, mas tem, porque ele tem uma inflamação sistêmica, mas do ponto de vista tecidual não tem. A pele não lesional, ela é
É,
idêntica em celularidade, inclusive, que uma pele saudável.
entendi. É porque você tinha mencionado fibroblasto em algum momento, e eu fiquei muito curiosa para saber se eles iriam mostrar, mas também não sei se você olhou especialmente os fibroblastos do tecido, vamos
Olhamos,
dizer,
olhamos.
paciente. Olhar.
Olhamos. Esse é um trabalho da Luciana Covre, doutora Luciana Covre, que é uma pesquisadora associada ao meu grupo. Ela tem olhado o estroma. O foco da pesquisa dela é justamente as alterações do estroma ligado à senescência, né? Então, ela
Ah,
tem um olhar especial para o fibroblasto. O que eu posso te mostrar desses dados preliminares, ou te falar dos dados preliminares, é que quando a gente isola o fibroblasto da lesão, ele já sai da lesão totalmente senescente. E senescência de fibroblasto é muito bem caracterizada, né? Começou com o Rei Flink na década de 60. Então, assim, ele não prolifera, ele expressa P16, P21, P38, ele secreta o SASP pra caramba, assim, ele é muito mais inflamatório do que um fibroblasto isolado de uma biópsia saudável, por exemplo. Então, ele tem todas as características de senescência e ele induz senescência de forma parácrina. Então, quando a gente co-cultiva esse fibroblasto com um linfócito que não é senescente, por exemplo, o linfócito fica senescente, né? Então, o que a gente acredita é que potencialmente, né, a inflamação iniciada pela infecção, né, vai induzir uma senescência no tecido e essa senescência autoalimenta a senescência de todo mundo que está lá mantendo aquele ambiente inflamatório crônico. O que a gente
Não,
está
mas é
tentando...
porque é um efeito local, né? A coisa dissemina para as células estromais essa informação, né? Que muda tudo, não tá. Dissemina é uma palavra meio engraçada, mas não altera de forma mais sistêmica as células estromais de forma global, né? Interessantíssimo.
Não, só local. Então é uma
Colocar
coisa local, só local. A
ali.
gente tem biópsia desses pacientes no membro contralateral e a gente tem também biópsias de pacientes que tiveram cura clínica. Então a gente vai e biopsia próximo da onde foi tirada a primeira biópsia da lesão e volta normalmente. Depois que você resolve o problema ali, a celularidade também fica idêntica. Então
Bom, né?
são dois pontos. Muito bom, que bom, né?
Muito bom, muito bom. Obrigada, Daniel.
Falou,
Pessoal,
Cris.
acho que tem mais um. Tem alguém que acho que o Douglas está aqui
Douglas.
para perguntar. E eu
Isso.
vou precisar ir embora, Daniel, que eu tenho aula agora às 2, tá? Então,
Beleza,
beijão aí
Gustavo,
para
cara.
você,
Obrigado aí,
parabéns.
cara. Obrigado,
Tá
viu? Obrigado pela presença.
maravilhoso, cara. Pena que a gente não tá fazendo assim pessoalmente, mas a
Ah,
hora que
pô,
puder a gente vai trazer aqui
com
também,
certeza, fico feliz,
tá?
viu? Obrigado pelo
Então tá bom,
convite. É sempre...
valeu. Abraço aí,
Valeu.
galera. Boa semana para todos.
Valeu, Gustavo. Douglas, quer falar?
Olá, Daniel. Parabéns pela apresentação. É mais uma curiosidade, na verdade. O caminho para levar a senescência ele é bem descrito, mas existem evidências que mostram um caminho reverso, uma célula que ela é senescente, ela consegue desvoltar, dadas as características que ela tem, tanto de mudanças epigenéticas, também como encurtamento de telômeros?
Ó, cara, essa sua pergunta também é uma pergunta muito boa. É, é muito difícil você reverter a senescência como um todo, para fazer, por exemplo, aquela célula que está lá no estágio final de diferenciação voltar a ser naive. Isso não acontece, né? Porque é como se fosse um processo, uma linha do tempo para a própria célula. Então, ela saiu de naive, vira memória central, vira memória efetora, parará. Então, assim, você tem modificações, você tem edição, né, de DNA que impedem com que você reverta para que ela volte a ser uma célula regenerada, né? Se a gente pode dizer isso. Mas você pode melhorar algumas coisas. Você pode, por exemplo, melhorar vias de sinalização que dificultam proliferação, por exemplo. Então, você pode fazer com que a célula senescente prolifere um pouquinho melhor. Você pode diminuir a capacidade inflamatória dela, bloqueando determinadas vias de sinalização, tá? Então, você consegue atacar pontualmente algumas características, mas você não consegue voltar à fonte da juventude e fazer com que a célula se reestabeleça e vire uma célula jovem novamente. Por isso que as drogas, elas eliminam as células senescentes. Essas drogas senolíticas, elas têm como função matar mesmo, tirar a célula senescente da jogada e não reverter as disfuncionalidades, tá?
Entendi, muito obrigado.
Tem uma última pergunta aqui, Daniel, no chat do Ian. Vou ler aqui. Fiquei um pouco em dúvida agora. Esse perfil senescente pode ser atribuído a uma resposta sistêmica à lesão ou é principalmente um fenômeno mais restrito ao tecido lesionado? Eu estava refletindo no primeiro ponto, até pretendia perguntar se viram algo sobre autoanticorpos pelo perfil imune muito destacado e as citações sobre a autoimunidade na discussão. E te agradeceu pela palestra.
Ô, Ian, é o seguinte, cara, o que acontece? Vou te responder aqui como esquartejador, né, por partes. Em relação à questão sistêmica, a gente mostrou que tem uma resposta sistêmica, mas ela é muito menor, mas é perceptível. Quando você compara as frequências de células terminalmente diferenciadas em pacientes versus controle saudável, né? Não gosto desse termo, não, controle saudável, não, voluntário saudável, para as nossas experiências e nas nossas mãos, como o paciente ele tem uma produção sistêmica de mediadores inflamatórios, isso é bem descrito, inclusive por outros grupos, ele tem TNF-alfa a mais, você consegue detectar interferon gama em alguns pacientes no soro deles, eles têm aumento de proteína C reativa, eles têm um tanto de marcadores inflamatórios, e isso, de certo modo, está associado com a promoção e diferenciação de populações para senescência. E isso consegue ser perceptível, né, principalmente no compartimento de CD8. Mas quando você vai na pele, cara, na pele só tem isso. Na pele você não consegue ver populações menos diferenciadas. Por exemplo, população naive lá, linfócito naive, você não acha na pele. Você acha muito mais efetor. Até central é muito pouco a frequência. Então, é muito mais lesional, apesar de ter reflexos sistêmicos bem tênues, mas tem, tá? Em relação a anticorpo, cara, eu nunca vi nada, eu nunca medi, não estou falando que não tenha, tá? Eu não vi porque eu não fiz. Mas é um outro ponto importante a questão de senescência de célula B, que tem outros impactos. Como o nosso foco é patogênese, e apesar de anticorpo ter um papel relevante na patogênese de algumas formas clínicas, é, a gente não olhou célula B considerando as características diretas de células que levariam à patogênese, que é a citotoxicidade, inflamação. Apesar de linfócito B ser uma boa célula inflamatória também. Então, cenas de um capítulo futuro, né? A gente já está todo enrolado aqui tentando fechar todos os braços abertos, aí o pessoal fala assim, não, mas e tal coisa? Cara, não dá, não tem gente. Se vocês não querem vir para cá, se vocês quiserem vir para cá, eu fico muito à vontade de estender mais análise, tá?
Mais alguma pergunta? Bom, o Anderson está te elogiando aqui, seu trabalho no chat, tem no YouTube a Marcela também te elogiou, enfim, vários elogios, Daniel. Mais alguém quer fazer alguma pergunta para o Daniel ou a gente libera ele? Deixa eu te liberar então, Daniel. Te agradeço muito, tá, pela disponibilidade. Parabéns pelo trabalho aí e fique à vontade aí para se juntar a nós aí nessa iniciativa do webinars toda segunda-feira, tá, Daniel? Você está mais do que convidado.
Eventualmente eu passo, assim, dou uma passada. O meu grupo ele recebe direto as coisas, enfim, eu achei fantástico a iniciativa de vocês. Vocês estão realmente de parabéns, cara. Juntar esforços de diferentes pós-graduações, eu acho que a gente fica cada vez fechado no nosso mundinho, né? Eu acho que ter essa possibilidade de discutir com outros grupos é fantástico. Eu ouso dizer que e no futuro breve eu vou tentar vincular a nossa aí a essa bagunça de vocês, viu? Mas
Não,
enfim,
vamos, assim, a gente tem que dar os créditos aí ao ICB da USP lá e ao Carlos Chagas, Instituto Carlos Chagas lá no Rio de Janeiro, que a iniciativa é deles, nós aqui como lá da regional da SBI, que você conhece, né? A gente está apoiando, e aí o nosso programa de pós-graduação também entrou para ajudar um pouquinho, mas assim, acho que a iniciativa é deles, a gente quer realmente,
fantástico,
quanto mais
cara.
que a gente somar esforços, melhor, né?
Fantástico, fantástico, viu? Vocês estão de parabéns, viu? E obrigado, viu, Thiago? Obrigado aos organizadores aí da Biofísica, do ICB. Fico muito feliz de poder apresentar os dados, enfim. Obrigado mesmo.
Maravilha. Colegas, então vamos encerrando por aqui. Até a semana que vem, segunda-feira, a gente se encontra novamente, tá? Daniel, obrigado de novo. Até mais.
Valeu, Daniel. Um abraço, cara.
Valeu, um abraço.